Hi there, welcome to another episode of Catch This, a show about viruses, immunology, vaccines, and lots about this pandemic. This episode is about a really important topic, so I recorded it in 3 different languages: English, German, and Romanian. If you happen to watch it in Romanian, I apologize. That is my weakest language. Today I will talk about SARS-CoV-2 vaccines. Are they safe? Do they really work? And what do we know about long-term side effects? I hope to answer these questions today, and please share with your German and Romanian-speaking friends. We have been in this pandemic for a full year now, and sometimes it seems that it will never end. When we were promised a vaccine a year ago, I had my doubts, and for good reason. I am, after all, a trained virologist with some experience in vaccine science. And to be honest, I was pretty pessimistic. It usually takes years, and often decades, before a drug or vaccine is approved by the FDA. Most of the time, it's not that easy. After 60 years of research for RSV vaccine development, we still don't have one. 40 years of research for an HIV vaccine, nothing. Herpes simplex, hepatitis C, etc., nothing. Each of these infections has its own challenges and complications in terms of making a vaccine. Why was it so easy for this vaccine? Did we already have a good foundation of basic research in place so that we could simply build on that and quickly make a new vaccine? Is SARS-CoV-2 not as complicated as other viruses so that maybe it was easier to make a vaccine against this pathogen? Or were we simply lucky? Those are all good questions, and there are good answers. Let's start with the mRNA vaccines. What is in these vaccines and how exactly do they work? There are currently 2 of this kind. One was developed by Pfizer in collaboration with BioNTech, and the other one by Moderna. These vaccines are not infectious and they do not integrate into our genome, meaning that there is no risk of infection or mutations in our genome that could be caused by the vaccine RNA sequences. The vaccine consists of mRNA, which encodes the SARS-CoV-2 spike protein, one of the proteins on the surface of the virus. This protein is very immunogenic, meaning that it induces a good immune response in humans. Once this vaccine is delivered into your cell proteins use this vaccine mRNA as a message to build the SARS-CoV-2 spike protein, which then elicits an immune response, usually in the form of antibodies and T cells. If your body is then later infected with the real virus, your immune system can recognize the spike protein on the surface of the virus and can induce a strong response. The vaccines made by Pfizer, BioNTech, and Moderna are such mRNA vaccines. But RNA is very unstable, and it is very easily degraded once it enters your body, so it has to be protected somehow. The Pfizer and Moderna vaccines are encapsulated in lipid nanoparticles, and they end up looking like these little vesicles, which protect the mRNA from degradation and also help improve uptake into cells. This way you get increased expression of the mRNA. The particle can fuse with our cell membrane, and this allows the inside, the RNA, to be delivered into the cytoplasm of our cells. Addition of cholesterol molecules makes the particle more fluid, and it probably also helps improve its fusion with our cell membranes. Polyethylene glycol, denoted here as PEG, has different functions. It increases the efficacy of the vaccine by hiding it from the immune system. It makes the particle more water-soluble because it consists of many hydrophobic parts, and it slows down the degradation of the particle because it does degrade after a while. mRNA vaccines are new in the sense that there has never been one that was approved for use in humans, but the technology and the concept itself are not new. Reports of the first successful translation of a foreign mRNA in animals were published in 1990, and this technology has been refined ever since. Progress in the field has been hampered mainly because both scientists and investors were concerned that the inherent instability of RNA could prevent its use for delivery as a therapeutic or vaccine. Anybody who has ever worked with RNA in a lab knows how fickle it is and how frustrating it can be to handle it. However, research in the past 30 years has shown that the stability of RNA can be increased through various modifications and delivery methods. One of the reasons why SARS-CoV-2 mRNA vaccines could be produced so quickly is because all of this basic science was already in place at the start of the pandemic. And although the SARS-CoV-2 vaccines are the first mRNA vaccines to be authorized for use in humans, several mRNA vaccines have undergone clinical trials in humans before, for at least 4 infectious diseases: rabies, influenza, cytomegalovirus infection, and Zika virus infection. Many people are worried that the foreign vaccine mRNA could integrate into our genome, like HIV, for example. But that is actually not possible, or shall we say, very unlikely. When HIV enters our cells, its genome is not immediately translated into protein. It is converted to DNA instead, and then that goes to the nucleus, where it is integrated into our genome. The synthesis of DNA from the HIV RNA genome happens with the help of an enzyme called reverse transcriptase. The integration into our genome happens with the help of an enzyme called integrase. Both enzymes, reverse transcriptase and integrase, are encoded by HIV. That means that HIV contains the message to make these proteins. The sequence of events that occurs once SARS-CoV-2 mRNA enters our cells is completely different. Both the vaccine mRNA and the protein that is made from the mRNA only stay in our cells for a pretty short time. The mRNA sticks around long enough so that it becomes translated into the protein, and the protein sticks around long enough so that it can induce production of antibodies. Then after a while, they degrade. Unlike with HIV, there is no reverse transcriptase to make DNA from the vaccine mRNA, and since there is no resulting DNA, it does not go to the nucleus. And even if this did happen, there is no integrase enzyme to integrate the DNA into our genome. Even so, some people have been concerned that the foreign DNA can still insert into our genome using line 1 elements. Line 1 stands for long interspersed nuclear element 1. Lines are regions in our genome that can transpose, or they can move themselves to different places in our genome. And certain portions of line 1s encode a protein that has reverse transcriptase activity. So the concern is that the vaccine mRNA could hijack these reverse transcriptase and insertion functions of line 1 elements and thus be inserted into our DNA. However, there are plenty of data showing that line 1s preferentially move their own genome, and this is just one example of a published paper showing that. There is no reason to think that a foreign novel RNA would have an advantage over what is already present in the genome and be reverse transcribed and inserted by these line 1 elements. Long story short, any concerns about the vaccine changing our DNA or becoming a permanent part of our genome are completely unfounded. Okay, so these are the mRNA vaccines, but then there are also the vectored vaccines. Let's take a closer look at those now. Currently, there are 2 that are in use, one from AstraZeneca and one from Johnson & Johnson. For vectored vaccines, you need a virus as a vehicle to get the vaccine DNA into our cells. Usually adenoviruses are used. These are viruses that cause the common cold. The vaccine consists of an adenovirus vector with an inserted gene that codes for the SARS-CoV-2 spike protein, the same protein that is used for the mRNA vaccines. The notion of using a virus as a vector to deliver vaccines to humans is based on the ability of viruses to enter cells by attaching to host cell receptors and releasing their genome into the cell. Upon injection into a vaccine recipient, the vaccine vector should enter cells and serve as a code for host proteins to synthesize the SARS-CoV-2 spike protein from the inserted gene. Ideally, the spike protein will then act as an antigen to prime the immune system to recognize SARS-CoV-2 if it infects the body at a later time. Adenoviruses are particularly suitable as vectors for delivering foreign genes into cells because they have a double-stranded DNA genome that can accommodate large segments of foreign DNA, and because they infect most cell types without integrating into the host genome. However, due to the prevalence of adenovirus infections in humans, they do cause the common cold, as I already mentioned. Most people already have adenovirus-specific antibodies that could bind and neutralize the vector itself. And this could make the vector less effective at stimulating antibodies to the foreign gene product. To get around this problem, the AstraZeneca investigators used an adenovirus of chimpanzee origin that does not normally infect humans. So recipients of this vaccine vector would not likely have pre-existing antibodies to the adenovirus vector itself. The adenovirus used to make the Johnson & Johnson vaccine Adenovirus 26 is of human origin. However, when tested, most people have very few antibodies that inactivate this adenovirus compared to antibodies against other adenoviruses. Therefore, potential recipients of the Johnson & Johnson vaccine are less likely to have pre-existing antibodies to the adenovirus vector itself. To further optimize the adenovirus vaccine vectors, both AstraZeneca and Johnson & Johnson investigators also deleted 2 genes. One that regulates viral replication. This ensured that the virus vector cannot cause an infection in human cells, and an additional gene to make more room inside the vector for the inserted spike gene. So that's how these vaccines work. All of them were tested in clinical trials, each in tens of thousands of people, and they are all very, very effective, much more effective than expected. But how exactly do the clinical trials work, and can we trust them? Clinical trials, particularly in the 3rd phase, are randomized, double-blinded, and placebo-controlled. What exactly does that mean? Randomizing means that participants were randomly assigned to the experimental group receiving the actual vaccine or the control group, so that the only expected differences between the experimental and control groups are the outcome variables studied, which are safety and efficacy. Randomizing trial participants eliminates unwanted effects that have nothing to do with the variables being analyzed. Double-blinding means that neither the investigators nor the subjects know who is receiving a particular treatment, the actual vaccine or the placebo control. Double-blinding leads to more authentic conclusions because they reduce researcher bias. Placebo-controlled means that one group of participants receives a placebo, a substance that has no therapeutic effect, so that we can be sure that the effects observed in the actual vaccine recipients are real and result from vaccination. All the SARS-CoV-2 vaccines were tested in such clinical trials, without exception, including the AstraZeneca vaccine. Use of this vaccine is currently on hold because out of 17 million vaccine recipients, 37 people developed blood clots at some point after vaccination. However, according to Louis Sheng, a molecular virologist at Cornell University, these numbers are still below the expected normal incidence of blood clots for thrombosis in a normal population. He reasons that what is worrisome is the fact that suspending vaccinations may actually result in an increase in total thrombosis cases. Thrombosis is common in serious COVID-19 cases, which is far more frequent in susceptible populations than the reported frequency of blood clots among the vaccinated. And it is really important to keep in mind that we don't have any evidence that the vaccine causes blood clots. The occurrence of blood clots in connection with vaccination is nothing more than a correlation, and correlation does not equal causation. There is no evidence that the AstraZeneca vaccine causes blood clots. So now for the important question. How did we end up with not just one, but several highly effective vaccines in less than 1 year? Because there are actually more SARS-CoV-2 vaccines than I described here today. There are several reasons. The most important is probably because we had a very good foundation of basic research. 30 years of research for mRNA vaccines. Conventional vaccine strategies involve attenuated or inactivated viruses, and these methods are very time-consuming. mRNA vaccines can be made more quickly because they obviate the need for growing and or repeatedly passaging viruses in cell culture, which can take a pretty long time. An additional reason, after many years of research with other coronaviruses, because we know of 6 others that infect humans, scientists knew that the spike protein was the important immunogenic protein. And in my opinion, we also had quite a bit of luck, because it could have turned out that the spike protein is not the important immunogenic protein, or that it has slightly different properties in this particular virus. And one last point, you know the maxim, good, fast, or cheap, pick 2. In this instance, we picked 2 all right, and this undertaking was anything but cheap. In short, vaccines are one of the biggest success stories of modern medicine. The World Health Organization estimates that vaccines save 2 to 3 million lives every year and prevent countless other cases of disease and hospitalization. Thanks again for listening. My name is Trudy Ray. Please subscribe.